So, oh, wait. Sorry, we're testing. But yeah, this is the highlight of the whole talk. So remember that. All right, so let's see. Okay, thanks, Tracy. Thanks for having me here to share our research with you, which is really aggression focused. I'm really excited to share with a group of scientist audience who share the same passion. So what we'll be focusing on today is, as the title stated, the changing part of aggression. But let's just say a little bit about aggression as a behavior, the background of it. So aggression, we consider it as, and I consider it as innate behavior. It's a stereotypical behavior, and it's observed widely across a wide range of species. So the reason we think it's innate basically means born with. So so we see it in these animals that they can be, isolated. They don't need to sort of learn it explicitly, but when the circumstances arise, they will be able to express those behaviors, from insects all the way to humans. So this behavior is very essential. It helps to secure the resource, to compete for mating opportunity, to defend oneself. So with all these important functions, that's why this behavior is existing and developmentally sort of hardwired. There are regions dedicated for it. And because of that, scientists have been searching for those regions for many decades, in fact, nearly a century. So in the early days, the experiments were done in cats, and it was found that this electric stimulation of the cat hypothalamus can evoke this dramatic behavior changes. So when the median hypothalamus is activated, it causes this defensive rage behavior, which is, you see the cat here is hissing towards the rat. And if the lateral hypothalamus is activated, it causes this predatory attacking behavior, so sort of a cold blood killing. And that sort of being the first clue, together with some lesion experiment, saying that hypothalamus is a very crucial region for the expression of those behaviors. And then now, fast forward for 70 years, so, but it's still 10 years ago, that basically summarized the findings I made when I was a postdoc fellow at the Caltech in Dr. David Anderson's lab. So at the time, we identified the ventrolateral part of the ventral median hypothalamus being a very critical part for aggression. So nowadays, we use mice instead of a cat as the model species. But nevertheless, there are actually hints of evidence across the literature saying that this region is conserved across many species, including primates. So what we found in that study is that when we inactivate these regions, then the mice start to become really peaceful. So the mice, the aggressive behaviors in the mice are actually quite readily inducible, especially in the males. So when the males are being single-housed and then you introduce another male intruder, then they will start to actually attacking each other. And that's called a resident intruder assay, and it considered territory aggression. So when we activated these regions, dramatically, we see this illicit acute increase of the aggressive behaviors. So it's not only against other conspecifics, such as another male, another female, but also it actually can happen against this inanimate object. In this case, it's a blown up glove. It was actually one of my lab members saying, hey, would that bite a human hand? I said, let's try it, but I don't want to put my hands there, which seemed to be a good idea. So this set of experiments really established that the VMHVL being a very critical part for the intermale aggression in the mice. And there are a series of studies that follow up on that, which really establish its indispensable roles now. So now I want to talk a little bit about not just identify the area, but also how these areas might change. So that sort of stem from the observation that aggression, despite the fact that it's innate, it's actually not a fixed behavior. It's a very flexible behavior that can influence a lot by the social experience. And most dramatically, it can be influenced by the winning and the losing experience. And this is so widely observed that it actually has a term to describe that, which is called the loser effect and the winner effect. The loser effect means that when the animal gets defeated, then they start to fight less. And when the animal wins, they start to fight more, and they actually tend to win more. So these are sort of histologically actually really important behavior changes, because every time you fight with an conspecific, it's an opportunity to evaluate sort of your resource holding potential compared to your peers. And if you have a higher resource holding potential, then engage in those behavior more, and then you can secure more resources. But if you are not good at it and you lose the fight because there is a bigger risk and a cost to it, then better to stay away from it. So this behavior changes is an important adaptation. But the question is, okay, so actually here's some videos just to see, to illustrate that point in the mice. So this is a naive mice, and you can see that this mice, when we put in the intruder here, very much interested in intruder. So intensively investigated intruders back and forth, and for a good 10-20 minutes. So I actually sped it up, otherwise we'll be spending a big chunk of the talk just looking at this video. But then after 10 minutes, just after a couple seconds, you will see the mice initiate the first bite onto this intruder here. So it's happening now. So once it starts to bite, then it will bite more frequently. So this is the naive animal. Now, this is the same animal actually, but after it winged repeatedly. And you can see the moment the intruder comes in, actually start to attack right away. So that is showing this winning effect. Basically, the readiness to attack increase dramatically with the winning experience. And then in the defeated animal, on the other hand, you can see that this mice, when we put in an intruder here, it actually tries to stay away from an intruder. So this intruder is is a benign intruder, so it's a non-aggressive mouse. So this intruder itself, it actually not impose a threat. It doesn't initiate attack throughout the whole action period. But this mice, nevertheless, never initiate attack towards this defeated mouse. So the question we hope to address is how this winning and the losing experience is changing the aggressive behaviors. Presumably, this experience is changing the neural circuits supporting the aggressive behaviors, and as a result of that, the behavior is being changed. So, of course, since the VMH-VL is being a very critical part of the neural circuits that are mediating those behaviors, so we're asking, does the activity responses of the VMH-VL cells will change? So to address this question, what we did is to use optical fibers to record the VMH-VL cell activities. So here we acquire a bulk signal, not from individual cells, and we express GCAMP, which is a fluorescence sensor for the capsid, so, and which reports the cell activities in general. So we record it from those cells, and then we allow the animal to recover for 3 weeks, and then we will probe the sensory responses to the aggressor cues when the mice is hadafixed. So we'll hadafix the mice, and having this anesthetized mice running on this little platform and deliver to our recording mice. The reason for that is because, as the animal become more and more aggressive, you see the behavior change dramatically. So the investigatory behavior also changes. So we don't want this sort of a mixed behavior change and a response change mixed together, which makes it harder to distinguish what is the cause of the response changes. So that's why we fix the behavior and just focusing on the neuronal responses. And now after we probing the responses for 3 days, we then have the animals either win repeatedly or losing repeatedly. And then we were asking how does that change the neuronal responses in the VMH-VO, and at the end we'll probe the sensory response again. So here is the result. This is a pre-weaning. So pre-weaning, the VMH-VO cells actually respond quite similarly to male and female. This was in fact a surprising result for me because male mice, they attack really only the males, they don't attack females. And this region is important for aggression. So if they respond similarly to stimulus, how could the animals know that which one to attack? But nevertheless, that's a pre-weaning result. And we found that as the animal win more and more fights, we see a drastic change in the activity here. We see the responses to the males increase and the responses to the females actually gradually decrease, or at least it doesn't increase. And this is changing response not just because of the social interaction alone, because we do have some animals that never become aggressive. And in those animals, we do not notice a clear change in the responses to neither male nor the females. And also when the animals become defeated, on the other hand, we see initially the response is similar between the male and the female. And over the course of defeats, they actually remain similar. However, the overall response is decreased. So what we're seeing here, if I summarize it here quickly, is that after weaning, the response to the male is going up and the response to the female is slightly going down, while after defeat, the response to both male and female going down, and there is no differences if just after social interaction. And if we look at it sort of in a different perspective, that is comparing the response to the males and the females, we found that actually only after weaning, and which is the only group of mice showing aggression in these plots, that they show a biased responses towards the male in comparison to the female. So this shows that the VMH-VO activity is indeed changed with the winning and losing experience, and changing in a way that's allowing it to differentiate the male and the female responses more clearly. So how does the VMH-VO cell activity change? That's the next question we'll be trying to address here. So basically, what is the sort of a more deep down in the cell levels and the synapse levels of what has been changed? So we thought that there are 3 possibilities. One is that it's actually caused by the synaptic plasticity, which is the most famous form of a plasticity in the brain. And this could be because of the synapse between the upstream regions and the VMH-VO has been changed with the winning and losing experience. Alternatively, it could be that the VMH-VO cells themselves are changing. The excitability of the cells may have changed that cause the cells to respond more readily or less so. The last possibility is that the change actually doesn't really happen at the VMH-VO at all. It was an upstream region of the VMH-VO, and the VMH-VO just sort of inherited those changes and showing the response changes. So in order to address this question and differentiate those possibilities, we turn to the in vitro slice electrophysiology. So in this case, we have the ESR1 ZS green cells. I should mention that actually all the recordings are down on the VMH-VO ESR1 cells, which is the population that express estrogen receptors, and this population has been known previously that's essential for the aggressive behaviors. So that's a population we've been focusing on. So here we use the ESR1 ZS green, which is basically all the cells in that's expressing ESR1 will have a green fluorescence, so allows us to identify the cells more readily. So for these mice, we have 4 groups. We either have the control, which will be just a single house them for the entire 15 days, or we have the mice who will single house them for 4 days, and then have them going through repeated weaning or social interaction or the losing experience. At the end of it, we take the brains, take the slice, and record from the cells, and to see how the activities of the cells are. So first of all, we look at the synaptic transmissions after all this experience, and we notice no difference. So there's no change in the excitatory synaptic transmission. This is spontaneous EPSE. and there's no change in amplitude or no change in frequency with any of those experiments. And then we looked at the iPSC, which is the inhibitory synaptic transmission. We also didn't see any changes. However, when we look into the cell properties, so we start to notice some differences. First of all, we see the renal base in the losing animals is going up, and for the winning animals is going down. So what is renal base? Renal base is basically the minimum amount of current you need to inject into a cell in order to get a single spike for the periodic recording, which is like a second. So some cells, they could fire spontaneously at a higher rate. That's why you will need to inject some negative current to bring it down to actually make it fire only one spike. So this shows that basically how much spiking the cells are spiking, basically. So we're seeing that in the losing animals, we need to inject a more positive current in order to get the cell to fire. And in the winners, we actually need to inject some negative current to bring down the firing of the cells. And we also noticed that there is a difference in the resting membrane potential. The losing animals has a more hyperpolarized resting membrane potential, which make them harder to fire action potentials. And the winners shows a trend of depolarized membrane potential. So furthermore, when we look at this current firing relationship, we see a more clear difference here. So we inject a series current from the negative to the positive, and you can see in the naive animals, as expected, when we inject more currents, the cell fires more, and at some point they sort of plateaued, the firing no longer increase. And in the social animals, it's a very similar profile here. But in the winners, you may notice that there's actually, first of all, there's some rebound spiking, more rebound spiking. And the second of all, you see that the spikes is becoming more dense. So they actually plateau at higher levels. And then lastly, in the losers, we see a very different patterns here. Initially, the cell fires sort of just fine, but then the cells fire a bunch of spike and the magnitude becomes smaller and smaller. And then they have this elevated levels of a membrane potential, but they no longer fire spikes. So this is we call depolarization block. So, and this is just sort of a summarize here. With the winner animals, we see an elevation of the spiking abilities of the cells overall, and they can reach a higher maximum in general. And with the losing, we see that the cells cannot hold the action potential, the spiking rate, especially when the more currents are being injected. So this is saying that the cells, even if you're trying to drive it by injecting a lot of currents, which is equivalent to positive synaptic inputs, the cells doesn't fire at a high rate. Why the winning cells, they're very ready to fire and can reach higher maximums. So now we want to go further deep down into the mechanisms, saying that why the spiking activity has changed. This, how the cells spike, how readily they spike is determined by a bunch of ion channels. So I want to see what kind of ion channels might be responsible for those changes. So we have a good colleague just in the corner, Dr. Dick Chen, who is a very good biophysics. So it's like, maybe we should go ask Dick to see whether he can provide some clues in terms of how to look into the channels. So what he told us is that, well, you can look at the maximal rate of rise of an action potential as a function of the membrane potentials, as a function of takeoff of potentials, and that's give you a readout of the sodium channel. Indeed, the rising phase of the action potential is largely determined by the sodium currents. So how quickly it can rise is determined by how much sodium currents you have. And the sodium channels, its properties varies with the resting membrane potential. That's why this takeoff is important. So when we look at that, we see that there is a clear rightward shift in the winner animals. So what does it mean? So basically, when the takeoff potential is the same, you have a higher maximum rate of rise in the winner animals than the other animals, which is suggesting there is more sodium currents available in the winner animals. And that's indeed the case when we use the voltage clamp to confirm that result. So we clamped membrane potential of the cells to different levels, which allows us to measure the currents passing through it. And in this case, we see that indeed the sodium currents in the winners are significantly higher than the sodium currents in the other groups. So this is the maximum sodium currents we observe here, which is significantly higher in the winners. But one thing is that we actually don't see a huge decrease of the sodium currents in the losers in comparison, at least to the naive animals. So, but nevertheless, we see, when we do the current spiking rate curves, we see that the cells in the losers cannot really fire action potentials very well because they enter this deposition block. But there are other properties of the sodium channel. So one is that how quickly it can recover after it being opened. So once it's opened, it enters this inactivable state, and it takes some time for it to become activable again. And that time lapse, how quickly it can recover, actually really important, because the more quickly it can recover, then it can be reused more quickly to support more action potential. So to address this question, we basically vary sort of the intervals between two small voltage clamp protocols. So we step it from -80 when all the sodium channels are available, and then to the maximum rate that all the sodium channels are open to measure the amount of sodium currents, and then we vary the amount of times for the second pulse to occur. So this tells us how quickly and the amount of the sodium currents can be recruited again. So when the duration, when the interval between this is long, as shown the example here, you see that pretty much all the channels, all the groups that the sodium currents has recovered, largely to the for the second to the third, first is similar. However, when the interval is short, you can see in the loser animals that there are actually much less being recovered in comparison to the winner and the social animals. And then when we plot a bunch of cells all together, we see that indeed below 70 milliseconds, the losers has a significantly less recovery for the second pulse. So which is suggesting that in the loser animals, it actually takes longer time for the cells, for the currents, sodium channels to be able to reopen again, which limits how fast the action potential can occur. And indeed, this depolarization block really is the action potential becomes smaller and smaller, is reflecting that the sodium channels are being depleted. So the other property of the sodium channel is its voltage dependence. So basically at a given voltage, how much sodium channel is ready to be opened. So in order to ask this question, we do a pre-post stepping. So we basically stepping it to different level of membrane potential voltage for a short period of time, and then step it to the maximum voltage, the voltage that gives the maximum sodium current. So this tells us at different voltage how much the sodium currents are available. And you can see this is basically at the -120 milliwatts, all the sodium currents, sodium channels are going to be ready. And then now we're stepping it to -60. You can see in the losers, there is significantly smaller in comparison to the other conditions. So if we look at the overall populations, you can see that the curve of the losers looks different. And if we just focusing on -60 here, which is where the resting membrane potential is, which is where it really matters, there is a significant difference between the losers and the other animals. So in the losers, in the resting membrane potential, they're only actually 60% of the channels are available, are readily open. Can be readily open versus there's about 80%. So although the total sodium channels are not significantly different from the losers and the naive animals, but there is the amount actually available can be opened is significantly less. So now sodium currents is not the only currents that's important for the action potential generations. There's also potassium currents and calcium currents. So I'm just going to talk about those quickly. So So the potassium currents, so when we do voltage protocols to isolate the potassium currents and blocking the sodium channels, we see that actually when it reaches these stable levels, there is no difference across all these different condition groups. But we do notice that there is appear to be a little sort of uptick, and that seemed to be different across different groups. So this turned out to be an A-type current, which is a transient current, and that current the channel will open and then quickly close. So this current is important because it opens around where the action potential generation, the threshold is. So once it's opened, the potassium channel is a negative force, which will making the cell leaky and make it harder to reach action potential threshold. So to isolate this particular current, we use this pre-pulse protocols. So we step it to -30, which then this stepping is in fact activate the A-type current. And then you can see that it's being activated here, and then we step it again, it no longer contain this current no longer contain the A-type. So by subtracting them, we can isolate the A-type current. And then when we look at it, we find that indeed it's increased in the loser animals and in comparison to the other groups, which will make basically the loser animals, the cells become harder to fire. So now the last thing we looked at is the calcium currents, which not going into the detail, but just telling you that there is actually some differences here. And that difference is mainly there are two types of calcium currents, and the difference is mainly at the T-type, which is the type that's activated at a low membrane potential. So, and also important for the action potential generations. Here we see that consistent with the fact that winning animals are more ready to fire action potentials, there is higher amount of the T-type calcium currents in the winning animals, and the least in the loser animals. So all that, I do an interim summary. So what it says is that social experience can change the level of aggression quite dramatically. And that change is accompanied by the changes in the VMHVL responses to this aggression-provoking cues. VMHVL itself is a very important region for generating aggression. Simply by activating these areas, you can drive the animal to attack. And that changing responses appear to be largely due to the changes in the VMHVL cell excitabilities. And this changing excitability is due to changes in the ion channel compositions and properties. And of course, there are some molecular mediators in between, and that's something we're trying to figure out next. So we were pretty happy with this finding. You know, they are quite to make sense. But around the time we sort of wrapping up this data set, we actually noticed two papers that published about around the same time that's saying that there is actually the experience cause a change in the synaptic transmissions from the median amygdala to the hypothalamus, especially the VMHVL. So like, what's going on here? Because we did not find any synaptic plasticity here versus those papers, they actually report there is synaptic changes. But one thing that's quite different from those papers and our papers is that we have been focusing on the long-term winner effect. So we use the animals that win a good 10 days repeatedly. But for those papers, they have been focusing more on the short term, at maximum 5 days, and mostly is one day or even immediately after. So it's like, could that be the cause of the differences we observe that how the neural circuits is adapting to those behavior experience? So to address this question, we thought, okay, let's just try to see whether the short-term winning will have a different effect as the long-term winning. So this is the first, just to remind you, the data we had acquired, I showed you earlier. That is, in the naive and tending winner animals, there is no difference in the synaptic transmissions, the excitatory synaptic transmission. And then when we looked at animals which win for 5 days, so we also looked at for winning for 1 day, it's a similar result. And we found that there is a drastic increase in the synaptic transmissions. So it increased by a couple folds, in fact. And then if we look at the iPSC, the inhibitory synaptic transmission, we see there's also some differences here, but not as dramatic as the naive animals, as the EPSC. And for those for the 5 days, when we look at the intrinsic excitability of the cells, such as the renal base, we actually don't see a difference. The 10-day and the naive are different, but the 5-day and naive are not different. And when we're looking at the current and the spiking relationship, we see that in the 5-day winners, the excitability was not increased at all. In contrast, the 10-day winners has a drastic increase in the excitability here. So what this suggesting is that the cells appear to go through 2 stages of plasticity. So before the VMH-VO excitability, cell excitability change, there is in fact changes in the synaptic plasticity. And whether that change in the synaptic transmission is required for the changes in the excitability is sort of an interesting question, and it's something we hope to be able to understand in the future. So now, I've told you a lot about the VMH-VO, but obviously VMH-VO is not the only place in the brain regions that can drive these behaviors. It's embedded in a complicated network. And now, over the last decades, with works from many labs, we now have a more complete picture regarding the connectivities of this VMH-VO regions, related regions, as well as the importance of each of those regions for aggression. So these are sort of the regions that has been implicated to be relevant for the aggressive behaviors. So we want to ask the questions, is the plasticities happen really in the VMH-VO only, or it's actually widespread across the aggression circuit throughout all these different brain regions? So in order to address this question, we decided to adopt a way that allows us to simultaneously recording from many of the brain regions all at the same time. So we constructed optical fiber bundles that can insert it into the different brain regions all at the same time. It's a very small footprint and custom made that allows us to acquire the activities in all these brain regions as the animal behave. So this is a quick sort of a illustration of the brain regions we've been targeting. So we have 2 arrays and single fibers allows us to target certain regions simultaneously. And these are the brain regions we have selected largely based on their implications in social behaviors as well as their connectivities with the VMH-VO. So this is the mouse that with the fiber implantation on the head. And this is from the other end of the fiber bundle, so we can basically image these fiber bundles, and based on the fluorescence fluctuations of each of the channels, we know that the activity levels of a particular brain region. So these are the regions we have targeted, and then we injected the virus from all of them, and then record from them. So they include 5 regions in the hypothalamus, 5 regions in the amygdala and extended amygdala, and then also there is a striatum regions, in particular lateral septum, which has been implicated in controlling aggression, and PAG, midbrain regions, important for the execution aggression, and the ventral subiculum hippocampal regions for emotion regulation. And now here, we do in the studies notice that there are actually some heterogeneity across the anterior posterior parts of the brain regions. So at the end, we're actually needing to separate them out for each of the brain regions into anterior posterior part in order to really be able to combine the data, as otherwise they are not really similar and another shouldn't be combined. So For the behavior assay, it's very straightforward. All we do is to have the animal starting from naive and then getting them repeated exposure to a male intruder and then a female intruder, and then over days, and see how does the response of different regions change. So this is showing one example recording traces. You can see that the mice is initiating attacking towards this biopsy non-aggressive mouse. And all these channels are being recorded activities, and when the attack occur, there is a flurry of activity across many of the brain regions. So, and here is sort of one recording sessions from all these brain regions when the male comes in, and then they investigate it for some shorter period of time, and then they start to attack. So one thing we notice is that during both investigation and attack, there's actually activity increase across many of the brain regions. So during male investigation, the activity largely is limited in the hypothalamus and amygdala, not the regions outside of it. But during attack, in fact, nearly all the regions except the lateral septum, which is interesting because lateral septum is known to control, suppress attack, not promote attack. So this lack of response sort of become unique features across all the regions. But we see that there is a really synchronized high activities across many of the brain regions during aggressive behaviors. So beyond the attack and investigation, the highest activity rise we observe, in fact, is when the male comes in. So that's like the first encounter of this intruders that really cause a higher increase in activities, which potentially is causing some state changes. And if we look at just these responses, and then compared to the others, you can see that other responses become dolphin because this is so big. And the response is given higher in the hypothalamus than the amygdala. That's sort of one feature we have seen it quite consistently across animals. The hypothalamus tend to have this higher initial responses in comparison to the amygdala. And additionally, we see that the hypothalamus response tend to faster. So it rises up faster, it reach on the peak faster, and the amygdala sort of slowly ramping up as the animals interact with the other animals. So the hypothalamus response is bigger and faster for this initial encounter. So we just, the data is still being analyzed. There's a lot of detail into it in terms of how this whole network might interact with each other. But one thing to address the question we set out to answer is basically, does the brain regions, do we see this activity changes across the entire aggression circuit? Oh, it's only in particular brain regions such as VMHDL. So here is the non-aggressive mice, how it is responding during the entrance. We focus on the entrance response because this is sort of the biggest response, most noticeable one. And now we have the animal become aggressive after a repeated weaning experience. And we see that there is a clear increase in the entrance responses to the males in multiple hypothalamic regions here. And in contrast, the middle regions appear to remain largely similar in terms of the responses. And if we look at the response to the female, we found that this is in fact not a general increase to the response to the social cues, because the response to the female in fact does not change after the weaning experience. And if we look at focusing on sort of the discriminability of a region to the male and the female cues by calculating this discriminative index, we see that before the mice become aggressive, for this non-aggressive green ones, that's before they become aggressive, you see it's largely above zero or around zero, meaning that there is no clear preference towards the males. And when the males become aggressive, then we see multiple regions that become clearly biased towards the male cues. So this says that the differentiability of the cells in a particular region to the male cue and the female cues, in fact, is gradually acquired with the social experience. And that's largely occur in the hypothalamus and also amygdala, the PAG areas, but not so much in the regions outside of it, including amygdala, striatum, and hippocampus. So this sort of to wrapping it back to this model we have proposed, how the social experience is changing aggression. Now we think that VMHVL is one of the regions, but the hypothalamus, variation structures might be in general, in fact, very flexible areas that can mediate changes in the social behaviors by changing the responses of the cells themselves. And that could be involving both synaptic changes and the cell excitability changes. The cell excitability change is particularly a dramatic way of changing the cell responses, and possibly because hypothalamic cells, they often have a quite dedicated functions in a particular behaviors, so they can afford to go through that kind of whole cell changes. Versus if you think about cortical neurons or prefrontal cells, they are so multitasking, so it doesn't make sense to change the overall responses of the cells as a whole. So now, just going back to the theme of innate and flexible, what we're thinking is that social behaviors can go through multiple phases, and that there are several points of changing. So first of all, when the animal detects social cues, they first need to decide whether they want to approach the cues or not. And social cues are generally attractive, so the animals are innately attracted to it. And then they will investigate these cues closely and determine, identify what the nature of that cue. And then based on the identity of the conspecific stimulus, they will engage in different consumatory behaviors. So, but there are a couple points that can change. One is this transition from the detection to approach. Once they detect the cues, do I approach? Do I approach quickly? Do I not approach at all? So with the defeat experience, in fact, the animals are going, switch the behavior completely. They no longer approach, instead of avoiding it. So there is one important point of change here from the detection to the approach. The other point of change is the probability of a transition from investigation to consumatory behaviors. So once I identify these individuals, what should I do? Do I engage in attack or not? Do I engage in mating or not? And that is somewhat dependent on the cell properties. We'll talk about the VMHVLs. So these cells which drive those consumatory behaviors and responding to those cues, male cues, and the higher they respond, then this transition will become more efficient. And then lastly, and if they, with the defeat experience, then this transition, the cells couldn't fire high enough, and then there will be no clear behavior output. And the last is that with the repeated weaning, with repeated experience, we also see evidence that the animals not only attack more quickly, but they actually attack more efficiently. They're able to take on an individual that's actually bigger in size. So this is sort of like me punch somebody who's really clumsy doing it, or a martial artist who can really fight very efficiently. So there is potentially a model learning component as well in the innate behaviors. Also, this has been not sort of a focus of research yet. So, but potentially there will be more research along these lines in the future. So with that, I would say innate social behaviors, they're innate, but they are actually highly flexible. There is multiple points in the circuit, in the behaviors that can change. And these are, I see them as very important and interesting questions. So lastly, I want to thank my lab members who are really the driving force for all this research, and in particular, Zhichao and Dayu, who are the two persons that carry out the experiments I have talked about today. So with that, I thank you very much for your attention, and remember to come back